Welcome, everyone. Today, we are going to explore the fascinating and frankly, slightly terrifying world of Taenia solium, commonly known as the pork tapeworm. Imagine hosting a tiny, uninvited guest that sneaks into your body through a piece of undercooked meat and decides to live in your digestive system for years. This parasite is like a traveler that absolutely requires two different environments to complete its journey. By understanding this incredible two-part journey, we can learn exactly how to break the chain and protect ourselves from infection. Let us dive in. Now that we know what our topic is, let us look at how this parasite survives. In scientific terms, Taenia solium is a digenetic endoparasite. This simply means its life cycle requires two distinct hosts to be completed. It cannot survive its whole life in just one place. The primary host is the human. This is where the parasite reaches sexual maturity and reproduces, specifically inside the small intestine. On the other hand, the secondary or intermediate host is the pig. The pig harbors the larval stage of the parasite within its muscle tissues, waiting patiently for the next phase of its life. Building on this two-host system, let us examine the physical forms the parasite takes. The life cycle involves several distinct morphological stages. We have the adult tapeworm, the egg, the six-hooked oncosphere larva, and the infective Cysticercus larva, which is also known as a bladder worm. When a human consumes raw or undercooked pork containing this Cysticercus, the larva's head, called a scolex, attaches firmly to the intestinal wall. From there, it begins growing into a long adult tapeworm, thus completing the cycle. Understanding these stages is crucial for identifying how the infection spreads. This brings us to a critical public health issue. What happens if humans accidentally ingest the eggs directly from contaminated food or water rather than eating meat containing the larvae? In this scenario, the larvae can hatch in the stomach and migrate to the human brain. This causes a severe and potentially fatal condition called neurocysticercosis. It is actually one of the leading causes of acquired epilepsy worldwide. By mapping out every step of the tapeworm's life cycle, doctors and public health officials can design effective interventions, like better sewage treatment, to prevent this debilitating disease. To truly appreciate how easily this parasite spreads and causes such diseases, we must look at its staggering reproductive capacity. The adult tapeworm is not just one solid body. It consists of hundreds of individual segments called proglottids. These segments mature as they move toward the tail of the worm. The mature ones at the very end are essentially just sacs filled with thousands of microscopic eggs. Because of this structure, a single tapeworm can produce an unbelievable number of eggs, contaminating the environment rapidly if proper sanitation is not maintained. Now that we understand the role of proglottids, let us look at how we quantify this biological output. We can calculate the total reproductive potential of a single tapeworm over a specific period using a simple multiplication model. The formula is RP equals NP times EP times D. Here, RP represents the total reproductive potential, which is the total number of eggs released into the environment. NP stands for the number of gravid or pregnant proglottids shed by the human host per day. EP represents the average number of eggs contained within a single proglottid. Finally, D represents the number of days the infection persists. If any of these variables increases, the total environmental contamination skyrockets. Before we move on, let us see this formula in action with a concrete example. Suppose a human is infected with a single tapeworm that sheds an average of 5 proglottids per day. Each proglottid contains approximately 40,000 eggs. We want to calculate the total eggs released over 1 year, which is 365 days. First, we substitute our known values into the equation. We multiply the 5 proglottids by 40,000 eggs to find the daily output, which gives us 200,000 eggs every single day. Then, we multiply that daily total by 365 days. The final answer is 73 million eggs per year. This astronomical number highlights why a single infected person poses a massive risk to an entire farming community. So, we have seen how many eggs an adult worm produces. This brings us to an important clinical question. How long does it take for a newly ingested parasite to start producing these eggs? This time frame is called the pre-patent period. It is the time between a human ingesting infected pork and the moment the adult tapeworm begins shedding eggs. This period includes an initial time for the Cysticercus scolex to evaginate and attach to the intestinal wall. After attachment, the worm grows at a steady rate until it reaches maturity. Understanding this timeline is crucial because it helps doctors know exactly when to test a patient's stool after a suspected exposure. To find the total pre-patent period, we first need to model the growth phase mathematically. We use a standard linear growth model applied to biology, which assumes the tapeworm grows at a constant rate once attached. The formula is T sub g equals L divided by g. Here, T sub g is the time taken for the tapeworm to grow to maturity, measured in days. L represents the target mature length of the tapeworm in centimeters. G represents the daily growth rate in centimeters per day. By dividing the total required length by the daily growth rate, we find exactly how many days the parasite spends purely focused on building its massive chain of proglottids. Now that we have our growth formula, let us calculate the growth time for a typical tapeworm. Let us assume the worm grows at a steady rate of 5 centimeters per day, and we want to find out how many days it takes to reach a mature length of 300 centimeters. We substitute our given values into the equation, dividing 300 by 5. The result of this division is 60. This tells us that the growth phase alone takes 60 days. During this time, the tapeworm is absorbing nutrients from the host's digested food, but is not yet reproducing. Having calculated the growth time, we can now determine the complete timeline from ingestion to reproduction. We do this using the total pre-patent period formula. T sub t equals Te plus Tg. In this equation, T sub t represents the total pre-patent period in days. Te is the initial attachment time, which is the number of days it takes for the scolex to firmly attach to the intestinal wall after ingestion. Tg is the growth phase time we just learned how to calculate. This simple addition accounts for every phase of the parasite's early development inside the human host. Let us finish our calculation to find the total pre-patent period. We will assume it takes an initial time of 3 days for the Cysticercus scolex to evaginate and attach to the intestinal wall. We already calculated our growth time to be 60 days. By substituting these values into our equation, we add 3 and 60. The total pre-patent period is calculated to be 63 days. This means if a person eats undercooked infected pork today, it will take approximately 2 months before tapeworm eggs can be detected in their stool. Testing too early will result in a false negative. Now that we have explored the math and the biology, let us look at the complete picture. Here we see the full life cycle diagram of Taenia solium. At the top is the human, the primary host, where the adult tapeworm lives in the intestine. Notice the arrows showing feces containing eggs passing into the environment. These are then ingested by the pig, our secondary host, at the bottom. Inside the pig, you can see the magnified muscle tissue harboring the dormant cysticercus, or bladder worm. Finally, the cycle completes when a human ingests this undercooked pork, bringing the parasite back to its primary host. This visual perfectly summarizes the predator-prey dynamic that sustains this parasite. To wrap up our lecture, let us review the key takeaways. First, Taenia solium is a complex parasite that relies entirely on a two-host journey between humans and pigs. Second, the adult tapeworm's ability to produce millions of eggs creates a massive environmental hazard, leading to dangerous conditions like neurocysticercosis if eggs are ingested directly. Third, understanding the mathematical models of its reproductive potential and pre-patent period is vital for medical diagnosis and epidemiology. Finally, and most importantly, by thoroughly cooking our meat and maintaining strict sanitation, we can easily break this cycle and protect global public health. Thank you for joining this exploration of the pork tapeworm.